Hari tu kita stop kat mana? Hari tu kita stop kat mana? Kita stop kat
Habis
mana?
bab 8 rasanya.
Habis.
Habis.
Habis bab 8.
Madam, bab 8 insulin tu.
Insulin tu cDNA dah.
Dah madam.
Dah madam.
Okay. Alright. Apa benda ni? Sekejap. Okay. Dah rupanya. Saya tak tahu sikit. Okay. Kita recap lah. Okay, kita recap on previous lesson. Saya dah suruh buat kerja ke?
Ada.
Soalan mana?
Soalan nombor 1.
Soalan nombor 1.
Biomix.
Okay. Soalan nombor 1, biomix.
1.
Alright, soalan nombor 1, biomix. Alright, figure 1 shows the cloning process in the production of a human insulin. Identify the mistake in the above diagram during the cloning process of human insulin. Okay, so what's the answer? Apa kesilapan daripada soalan tu? Apa silap dia? Okay, kita nak hasilkan human insulin. Bila kita nak hasilkan human insulin, kita kena extract apa? Right? When we want to isolate, we want to produce human insulin, what we going to isolate? Kita akan isolate apa?
mRNA.
Right? mRNA. So we're going to isolate mRNA. Okay, mRNA. So itu adalah mistake dia. Kita akan isolate mRNA. Kita tak isolate DNA. Okay, kita tak isolate DNA. Kita isolate mRNA. So the answer is the DNA from donor cell was isolated and used instead of mRNA. Okay, the DNA from donor cell was isolated and used instead of mRNA. Okay. So itu adalah jawapan dia lah. Okay, how to correct the mistake stated in A1? Macam mana nak betulkan? Macam mana nak betulkan? Macam mana nak betulkan? Macam mana nak kita nak betulkan mistake ni?
Madam nak try?
Boleh, sure.
DNA must undergo transcription to form mRNA.
Okay, DNA must undergo transcription. Ataupun awak boleh cakap, instead of, macam mana kita nak betulkan, sebab kita dah ambil DNA kan, daripada soalan ni kita dah ambil DNA. So macam mana kita nak betulkan adalah, kita akan isolate mRNA. Itu cara pertama untuk betulkan. Kita isolate mRNA from the donor cell. Okay. First benda yang awak kena betulkan adalah isolate mRNA from the donor cell. Macam tu. Okay. Alright, next. Kenapa kita kena isolate mRNA bukan DNA? Kenapa kita kena isolate mRNA, not the DNA? Kenapa bukan DNA? Why? Why not DNA?
Because Madam can advise.
Boleh, sure.
Because DNA contains intron.
Andai, okay, DNA contain intron. So bila dia contain intron, it cannot recognize by host cell. Okay, DNA contain intron. Okay, DNA contain intron, and this intron cannot recognize by host cell. Okay, itu adalah reason dia. Okay, intron cannot recognize by the host cell. Alright. Okay, state 3 of characteristics of plasmid. Okay, kalau karakteristik of plasmid, plasmid ni apa? Plasmid ni kategori sebagai apa dia punya tools, apa plasmid. Dalam tools apakah dia punya, okay.
Cloning vector.
Okay.
Vector.
DNA cloning vector. So plasmid duduk dekat sini. Okay, apa dia punya karakteristik? Ada berapa karakteristik dalam kita punya lecture note?
4.
4.
Empat. Okay, kita ada 4. Okay, kita ada 4 karakteristik. Okay, so awak boleh soalan 3 markah. So awak boleh letaklah. Ni, okay. Boleh letak 3. Okay, boleh letak 3. Okay, able to accept foreign DNA in multiple cloning site, able to replicate freely in the host cell because of the presence of origin of replication, possess selectable marker, okay, which is resistant to antibiotic. Sekejap eh. Okay, yang last kali able to express or amplify the clone genes. Alright. So kalau awak tengok soalan-soalan structure, dia selalu tanya characteristics. Okay. Next, what are the requirements for the formation of P? Apakah P? P tu apa? Dalam gambar P apa?
Recombinant DNA.
Recombinant DNA. Kalau saya tak nak guna recombinant DNA, apa perkataan saya boleh guna? Yang ni. Apa? Saya tak nak recombinant DNA. Apa jawapan saya boleh guna selain recombinant DNA?
Recombinant
Apa dia dekat situ?
plasmid.
Recombinant plasmid. So apa dia punya requirements? First requirement.
Madam?
Kita dah bincangkan soalan ni.
No,
Belum eh? Oh.
belum.
Okay, apa dia punya requirement?
They must be
Target
cut
DNA
by
ni.
the same restriction enzyme.
Very good. Cut using same restriction enzyme. Okay, bila cut using same restriction enzyme, they must have complementary, sticky or blunt ends. So this is the requirement. Okay, the target DNA have complementary, sticky or blunt ends. Okay. Second answer, the DNA fragment of the target DNA and the plasmid must be compatible. Okay, DNA fragment and the plasmid must be compatible. Right, that's the answer. Okay. Alright. So, saya akan explain lagi yang ni supaya dapat awak faham lagi lah. Okay, boleh ke saya explain lagi?
Boleh.
Okay. Alright. So, kita tak jumpa dah minggu ni. Okay, ni this is the first and last. Okay, we going to meet secara Google Meet. Okay, so these are the steps to produce cDNA for human insulin. Okay, first mRNA for human insulin is isolated from pancreatic cell. Kalau soalan cakap general, step to produce cDNA for human growth factor. Okay, human growth factor. So awak cakap lah mRNA for special cell, as mRNA for growth protein is isolated from specific cell. Okay, then the mRNA is used as template. Okay, and then it will undergo reverse transcription catalyzed by reverse transcriptase. Okay, catalyzed by reverse transcriptase. So this is the first enzyme that you're going to use. Okay, reverse transcriptase. So the second enzyme is the DNA polymerase. Okay, question might ask you what is the function of reverse transcriptase and also the function of DNA polymerase. Right? So mRNA used as template undergo reverse transcription catalyzed by reverse transcriptase. Why? To produce single-stranded cDNA. Apa maksud C kecil ni? Apa maksud huruf C tu?
Complementary.
Complementary. Okay. So kita punya purpose adalah untuk hasilkan double-stranded cDNA. So kita first we going to produce single-stranded dulu. And after that, the mRNA is degraded. Maksudnya dia akan dipecah-pecahkan lah. And then the single strand cDNA act as template. Then the DNA polymerase is added to catalyze the synthesis of second strand of cDNA to produce double-stranded cDNA. So in this process, apa nama proses ni? Kalau for this process number 2, we have reverse transcription. What is the process in step number 6? What is the process in the step number 6? For number 2, we have reverse transcription. Okay, what about number 6? 6 apa? Proses apa?
Replication.
Pandai, Syafira. Pandai. Siapa bagi tahu tadi? Manisha. Okay, very good. DNA replication. So if the question asks you to identify 2 steps, okay, so reverse transcription, reverse transcription, DNA replication. Okay, kalau dia tanya enzymes, reverse transcriptase and also DNA polymerase. Alright, so this is the process. Okay, this is the process that is yang ditunjukkan melalui diagram. Okay, that is shown using diagram. So we have mRNA as a template. We have the process reverse transcription, catalyzed by reverse transcriptase, catalyzed by catalyzed to produce single-stranded cDNA. And then mRNA will be degraded, and then single-stranded cDNA now act as template. Okay, to produce double-stranded cDNA, catalyzed DNA polymerase. Okay, itu adalah step untuk hasilkan insulin. Step to produce cDNA for human insulin. Alright, so now we're going to produce. Okay, production of human insulin. Kita nak hasilkan produk human insulin melalui 5 steps. Apa 5 steps tu? 5 steps tu apa? Apakah 5 steps in gene cloning? Apa dia? Asha. Aliana Najwa bagi satu. Aliana Najwa has left the meeting. Amira? Bagi satu Amira.
Isolation of DNA.
Okay, first isolation. Isolation of target DNA and the plasmid. Okay, tapi kalau dah soalan cakap production of human insulin, kita cakap isolate mRNA from human pancreatic cell. Okay, what is the second step? Diana, what is the second step? First we have isolation. Second step is?
Cut with the same restriction enzyme.
Cut with using same restriction enzyme. Very good. Number 3, step number 3. Carmila?
The cDNA is inserted into plasmid.
Okay, inserted. Okay, 3 adalah inserted. Saya nak step saja. Yes, insert. Okay, step number 4. Hana, step number 4.
Transformation and amplification.
Transformation and amplification. Number 5. The last one is Jihan.
Screening. Blue, white.
Screening. Blue white screening. Okay, very good. So katakanlah soalan cakap describe the production of human insulin. So awak kena spesifikkan lah. Isolate mRNA from human pancreatic cell. Then cerita mRNA is used as template for reverse transcription catalyzed by reverse transcriptase. Why? To produce single-stranded complementary DNA, cDNA. Then the DNA polymerase is added to synthesize second strand of cDNA to produce double-stranded cDNA. Okay, cerita spesifik. Sebab ini baru kita isolate mRNA. Lepas tu kita kena cerita macam mana mRNA ni nak tukar jadi double-stranded cDNA. Okay, sebab apa kita kena tukar kepada double-stranded cDNA? Kira kenapa kita kena tukar double-stranded cDNA? Kira kenapa kita kena tukar dengan double-stranded cDNA?
So that the complementary parts can be cut easily.
Okay, acceptable. What? Apa lagi? Anas, kenapa kita kena tukar kepada cDNA?
Sebab plasmid dia double stranded.
Pandai, itu jawapan saya nak. Itu point yang pertama. Thank you Anas. Sebab plasmid adalah double-stranded cDNA. Double-stranded DNA. Sebab itulah daripada mRNA, kita kena tukar kepada double-stranded cDNA. Supaya double-stranded dengan double-stranded compatible. Okay, compatible. Alright, sebab tu kita kena tukar. And then step number 4, cDNA and plasmid are cut with same restriction enzyme to produce complementary sticky ends. Okay, then step number 5, the cDNA. So notice that we don't use target DNA anymore. We use the term cDNA. We don't use the term target DNA or the gene of interest. Okay, sebab apa? Sebab kita punya produk sekarang ni adalah cDNA. So the cDNA is inserted into plasmid and joined using DNA ligase to form recombinant DNA. Recombinant DNA introduced into the host cell via transformation and then amplified into the host cell. Lastly, we have process screening. Okay, we use blue-white screening. Boleh ke? Kali ke berapa dah saya explain? Kali ketiga, keempat, kelima? Boleh ke? Kanak-kanak boleh ke? Boleh. Ada soalan nak tanya?
Boleh, madam.
Ada soalan ke nak tanya? Soalan tak? Ada soalan tak? Soalan tak? Ada soalan tak? Kanak-kanak ada soalan tak?
Tak ada madam.
Tak ada madam.
Oh my god.
Ada, madam.
Oh, saya dah nak pasang kuat. Tak ada? Alright, tak ada. Okay, kalau tak ada, betul tak ada soalan. Kalau tak ada soalan, nanti saya akan assign, saya tak assign lagi lah. Nanti dah start cuti macam esok lah kot. Esok saya akan assign dua video app puzzle and 2 quizzes. Satu quizzes daripada subtopik 8.1 dengan 8.2 kot. Ke saya gabungkan je. Tengoklah macam mana Okay, tengok saya gabungkan ke. Tengoklah macam mana Alright, so saya dah suruh buat saya nak awak buat kejap soalan. Okay, soalan number question number 7. Okay, boleh tak buat kejap dalam masa 5 minit? 7 minit lah, 7 minit. Okay, boleh ke? Boleh ke guys?
Boleh.
Soalan satu ni je, last. Okay, soalan nombor 7. Buat 7 minit. Kita bincang and then habis. Alright, 7 minit. So kita sambung balik ke 3:30. Okay, 3:30. Kak Rita. Kak Rita. Kak Rita. Siapa? Ha? Ya ke? Kesiannya. Astagfirullah. Calon. Water dengan aqueous solution sama?
Water sajalah.
Kalau
Solution
aqueous,
boleh kita larut dalam air.
oh, so lain lah dua benda tu kan? Alright, thank you.
Kalau kita water tu lalu kita rujuk pada pure water.
Oh, okay. Dah ke? Cepat sangat kot. Okay, nanti dah siapa belum angkat tangan? Kejap. Siapa belum angkat tangan? Okay, take your time. 3:30, 3:30. Dah ke? Siapa belum angkat tangan? Ya Allah, 4 markah je weh.
Dah.
4 markah 4 minit je. Okey, tak tahu. Kenapa tak tahu?
Dah.
Dah.
Okey, identify A. What is A? Apakah A?
mRNA.
mRNA.
mRNA. Pandai. Siapa aja pandai-pandai ni? mRNA, okay? mRNA. Alright, enzyme Y. What is enzyme Y? Mana enzyme Y ni? That catalyze the production of B. Where is enzyme Y?
Reverse
Apa?
transcriptase.
Reverse
transcriptase. Pandai, very good. Reverse transcriptase. C is produced with the aid of DNA polymerase. What is C? Apa C?
Double
Double-stranded
stranded.
Double-stranded cDNA.
Double-stranded DNA.
DNA, double-stranded DNA salah. Double-stranded cDNA. Okay, double-stranded cDNA. Okay, double-stranded DNA saja salah. Double-stranded cDNA. Okay, what is the role of DNA polymerase in this process?
To catalyze the synthesis of the second strand of cDNA to produce double-stranded cDNA.
Okay, itu jawapan dia.
cDNA.
Catalyze the synthesis of second strand of cDNA to produce double-stranded cDNA. Okay, so saya nak buat macam biasalah. Buat je semua biomix, structured question dan juga MCQ. Right, buat juga MCQ, multiple choice question. Ada berapa soalan untuk MCQ ni? Ada 40 soalan. Okay. So hari tak ada jumpa dah kita ni. So minggu depan kita masuk chapter 9. Boleh?
Boleh.
Okay, siapa yang dah ada kat college angkat tangan? Oh, ramai yang dah ada. Okay. So, okay ke? Line okay ke? Line internet okay ke?
Slow, madam.
Samalah kita. You are not alone, guys. You are not alone. Okay? Kan? Itulah nak buat macam mana. Ini dah termasukkan ke dalam lecture note? Yang ni?
Dah.
Dah. Alright. Okay, so awak kena buat apa cuti ni? Kita balik awak kena buat apa?
Add puzzle.
Awak kena buat apa? Hiking. Buat apa?
Puzzle, biomix, MCQ tu.
Lagi MCQ saja ke?
Semua biomix.
Semua biomix. Lepas tu saya akan bagi satu soalan lah. Okay, saya akan bagi satu. Sekejap. Caption pula ni. Saya akan bagi satu soalan essay dekat Google Doc. Okay, so